Hi friends, today's video is about Neisseria gonorrhea. So the Neisseria gonorrhea is a gram-negative, non-capsulated, non-motile, kidney-shaped diplococcus. So why it is called gram-negative? So if we are doing the gram staining procedure and we observe the smear under the microscope, we can see the bacteria stained pink color, which is actually the color of secondary stain or the counter stain. Hence it is called gram-negative. It is not covered by any capsule and it is also non-motile. Then here we have to remember kidney-shaped diplococcus. So the word coccus itself means the bacteria is spherical. Diplococcus means two bacteria adhere together, that is they are paired. Now it is kidney-shaped that you can see in this diagram clearly. So the adjacent surfaces are concave, hence it is kidney-shaped. The other species of Neisseria, that is Neisseria meningitis, it is lens-shaped. There we have the adjacent surfaces flattened. Yeah, like this. Okay, so this is kidney-shaped diplococcus. Remember that. And this bacteria can cause gonorrhea, which is a sexually transmitted disease or a venereal disease. Coming to the virulence factors, the Pili are the principal virulence factors. It helps the adhesion of the bacteria into the host cells and also prevent the bacteria from phagocytosis. So this is the Pili. Next we have the outer membrane proteins, mainly two. One is the protein 1 and the other is the protein 2. So the protein 1 is porins. So as if the name suggests, they will produce some pores or the transmembrane channels which help in exchange of molecules across the gonococcal surface. There are two main serotypes. One is pore B1A and the other is pore B1B. So the pore B1A can produce local as well as disseminated gonococcal infection. So we have to know the two terms. So I said this is a venereal disease. That means the infection is mainly in the genital region. So when it is confined to that genital region itself, it is known as local infection. Sometimes the bacteria may enter into the circulation and via the blood it can produce various other symptoms. Then it is called disseminated gonococcal infection. So the pore B1A can produce local as well as disseminated gonococcal infection. While the pore B1B, it usually produces only the genital infections. The second protein is protein 2, that is opacity associated protein, which help in adhesion to the neutrophils. So many other factors are there. To name some, we have the Lipo oligosaccharides. So I stressed on oligosaccharides, which means commonly in other gram-negative bacteria, we are having lipopolysaccharides as endotoxin, but here we have lipo-oligosaccharides. And the others are IgA proteases, transferrin binding proteins, lactoferrin binding proteins, and so on. Coming to the clinical manifestations, first in male, acute urethritis. This is the most common manifestation in males, and this is characterized by purulent urethral discharge. Okay, so you know how the word gonorrhea came? So actually, the gonorrhea is derived from flow of seed resembling semen. So as we can see in this diagram, the incubation period is about 2 to 7 days, and if it is left untreated, it can lead to various complications like epididymitis, prostatitis, etc. Now, the important word we have to remember here is Watercan perineum. The infections may sometimes spread to the urethral tissue, periurethral tissue, and causes abscess with sinus. So this abscess with sinus formation in the periurethral tissue is called Watercan perineum. Okay, remember Watercan perineum. In females, usually the infection is very less and they are usually asymptomatic carriers. Okay, it is less severe and they are usually asymptomatic carriers. There is some mucopurulent cervicitis with scanty vaginal discharge. So here you can see the scanty vaginal discharge, and there will be vulvovaginitis, so the inflammation of vagina. But this is usually rare in adults. Adult females, this is rare because of two reasons. One is the low pH, because we know the vaginal environment is usually acidic, that can kill the bacteria. And second one, the lining epithelium. It is stratified squamous epithelium, which can prevent the bacterial entry. However, this can occur in prepubertal as well as postmenopausal women because their vagina is lined by thinned out mucosa and there is also higher pH. Yeah. One more point I have to mention is the spread. Sometimes the infection spread to the Bartholin's gland, endometrium, fallopian tube, causing salpingitis, that is the inflammation of the fallopian tube, pelvic inflammatory disease, and this may lead to sterility in females. Next, a rare complication that is called Fitz-Hugh Curtis syndrome. So, there is peritonitis with associated perihepatic inflammation. Remember, this is peritoneum, not perineum, but peritoneum. So, there is inflammation of the peritoneum as well as inflammation around the liver, that is perihepatic inflammation. So, this is the characteristic image. Here, you can see this is actually the intraperitoneal exudates that cause violent string adhesion between the peritoneum and the capsule of the liver. This is Fitz-Hugh Curtis syndrome. In pregnant women, it can cause prolonged rupture of the membrane, chorioamnitis, premature delivery, and sepsis in the infant. In neonates, there is a disease called Ophthalmia Neonatorum. So, it is characterized by purulent eye discharge. It usually starts within 2 to 5 days of birth. Here, we have to remember 2 points. One, this infection occurs only at the time of birth and it is from the colonized maternal genital flora. We have heard about various infections that can spread via placenta, but this is not that case. This occurs only at the time of delivery when the baby comes in contact with the genital region of mother where the bacteria are colonized. These 2 points we must remember. Next, disseminated gonorrhea. So, there will be, it's a rare case, the bacteria entering into the blood and causing gonococcal bacteremia, which is characterized by polyarthritis, skin lesions, conjunctivitis, endocarditis, and so on. So, this is called disseminated gonorrhea. Next, the epidemiology. So, gonorrhea is an exclusively human disease, which means we cannot find this disease in any other animals. It is an exclusive human disease. And the source is usually female carriers because they are usually asymptomatic now. So, they cannot themselves identify they are infected with the bacteria. So, they may sometimes act as a source of infection. Sometimes the patient itself can also act as a source. Then the transmission, it's via two route. One, the sexual contact, and the other from mother to baby at the time of birth. Okay, so one point also remember, this infection, it is common in HIV infected persons. Okay, so the transmission is about 3 to 5 folds higher in HIV infected patients. Coming to the lab diagnosis. So, for any disease, the laboratory diagnosis, we have to follow a particular order. First, we have to mention the specimen, then we have to mention the microscopy, then culture microscopy if needed. Then identification via three methods. One is the biochemical method, the other is the automated, and the other is the molecular. Finally, the treatment and prophylaxis. So, this is the order. First one, specimen collection. So, in the gonorrhea, usually urethral swab in male and cervical swab in female is preferred. Okay, so one thing you remember, we should not use cotton swabs because the cotton can inhibit bacteria. So, the rayon or Dacron swabs are preferred. So, this is the procedure of taking a swab in male. Coming to the transport medium, actually the specimen should be transported immediately. If it is not possible, it should be collected with the charcoal coated swab and kept in transport mediums such as Stewart's medium. So, this is the Stewart's medium and charcoal contained medium called Amis medium. Currently, there are various commercial transport devices available such as Jambec or Gonopac system. So, this is the diagram of Jambec. Microscopy that we discussed in the first slide, it is gram negative. It is intracellular, usually found within the cells, kidney shaped diploid. Okay, it is highly specific and sensitive in men, but this is not about only 50% sensitive in females because the Neisseria species is usually a commensal in the female genital region. So there will be some confusions in the interpretation. So in the case of diagnosis of gonorrhea in females, we must do a culture. Okay, So the culture, we can use various medium. Chocolate agar can be used. However, the selective media are Thayer Martin medium. So you know what is a selective media? It will inhibit the other common cells and allow only the pathogenic bacteria to grow. So for the gonorrhea, we have the Thayer Martin medium. It is nothing but the chocolate agar mixed with three antibiotics. One, the vancomycin, colistin, nystatin. So vancomycin, it is used to inhibit the gram-positive bacteria. Then colistin, it can inhibit other gram-negative bacteria, and the nystatin, it is antifungal. So inhibit the fungal growth. Okay, so this is the Thayer Martin medium, and this is the after the culture, that is you can see the colonies of the bacteria. Also we have modified Thayer Martin medium, that is nothing but Thayer Martin medium added with trimethoprim. So this trimethoprim is used to inhibit the Proteus species. Okay, finally identification. So the first we have to mention biochemical. So it is oxidase positive. So you can see the purple color. You know how the oxidase test is performed? First we have to take a filter paper. It has to be soaked with an oxidase reagent that is called tetramethyl paraphenylene diamine dihydrochloride. Okay, tetramethyl paraphenylene diamine dihydrochloride. And then the filter paper soaked with this reagent is smeared with bacterial colony, and if this bacteria produces cytochrome oxidase enzyme, then this smeared area turns into purple color. So this gonorrhea, from this test we can confirm that gonorrhea will produce cytochrome oxidase, hence the purple color is formed. Next one, it is catalase positive, because you can see the effervescence. So how catalase is performed? Usually we will mix the hydrogen peroxide and the bacterial colony, and if that particular bacteria produces the catalase enzyme, it will produce effervescence like this. Okay, next it will ferment only glucose. So the automated method, Maltitov, and molecular method, that is the nucleic acid amplification test, like the PCR, can be used. Coming into the treatment, third generation cephalosporins are used. For example, ceftriaxone and cefixime. Prophylaxis. There is no vaccines available. Early detection of cases is found useful, and we have to treat both the partners, and we have to trace the contacts and treat them also, and we have to provide health education about safe sex practices. Okay, so finally, for the sake of completion, we have to mention about the non-gonococcal urethritis. So when we have some chronic urethritis and we cannot identify any gonococci, it is uh, designated as non-gonococcal urethritis. Okay, so there are various agents that can cause non-gonococcal urethritis, like bacteria, virus, fungi, parasites, and so on. And the main differences, it is in this tabular column. The main thing we have to remember is the onset. In the case of gonococcal urethritis, it's about 48 hours. Okay, where in the case of non-gonococcal urethritis, the symptom starts only after 1 week. That is the main factor we have to remember. So that's about Neisseria gonorrhea. Hope you understood, and thanks for watching. If you find the video useful, kindly like, comment, share, and please don't forget to subscribe. Thank you.